Good morning. Today we will discuss genomic imprinting and how, by doing this genomic imprinting, eukaryotic genes are regulated. Now, what is genomic imprinting? Most of the eukaryotes are diploid organisms. You would predict that it doesn't matter which allele of any given gene came from the mother or father. Given an example, we are having 23 pair of chromosome and there are so many genes. When we talk about a gene, we generally don't mention which copy we are talking about, either the maternal copy or the paternal copy. This is the general scenario. This is true in most cases, but there are important exceptions. The difference between the genes reside in how they are modified or imprinted differently in females and males. Now there are examples. When we are talking about the gene, we need to think now the maternal copy we are talking or the paternal copy we are discussing. In some cases, scientists have found that either the maternal copy is functional or the paternal copy is the functional. So one of them is functional, the other one is non-functional. How it is happening? Maybe some modification takes place. Now the evidence exists in mice and humans. Come to the in details of epigenetics. Genomic imprinting is the epigenetic phenomena. Now what is epigenetic in biology? Epigenetic is the study of heritable phenotype changes that do not involve alteration in DNA sequence. Now this imprinting is an epigenetic phenomena which is not following the typical Mendelian inheritance. And this imprinting by which certain genes are expressed in a parent of origin specific manner. That's what both the allele are not functioning here. One of the allele, either the maternal or the paternal allele is working and the other one is silenced or not functioning. How it is not functioning? That there is a mechanism. This phenomena we call genomic imprinting and this is an epigenetic phenomena. Genomic imprinting is an inheritance process independent of classical Mendelian inheritance. It is an epigenetic process that involves DNA methylation and histone methylation without altering the genetic sequence. Now, how this imprinting is happening, how we are informing that one allele is non-functioning and we are telling that it is imprinted. What is the mechanism behind it? Scientists have found most of the cases the allele is before that allele some specific area is methylation takes place or in the histone that is methylated and maybe it is very much tightly packed. That's why the expression, the transcription is not happening from that particular origin, from that particular allele. These epigenetic marks are established, that is imprinted in the germline, sperm or eggs of the parent and are maintained through mitotic cell division in the somatic cells of an organism. That means when it is one allele is imprinted in one individual, his or her sperm or the egg cells that is also imprinted and in the next generation also that allele will be imprinted. Now, if the allele inherited from father is imprinted, it is thereby silenced. That meaning imprinting, that word is meaning it is somehow stopped, not functioning. So, what is the mechanism? Just I mentioned the mechanism is methylation. Before the allele, the promoter sequence, there is a CpG island that may be methylated or the histones are tight, very lysine is methylated. So, lysine amino acid in the histone. So it is so much tightly packed. So heterokeratin formation takes place in that region. So if the allele inherited from father is imprinted, it is thereby silenced and only the allele from the mother is expressed. Now, if the allele from the mother is imprinted, what is the meaning? That means mother, that specific allele is not functioning, but the father's paternal allele is working, expressed. Forms of genomic imprinting have been demonstrated in fungi, plant, and animals. Genomic imprinting is a fairly rare phenomena in mammals. Most genes are not imprinted. So remember, most genes are not imprinted. Next discussion is that you need to know during development process, appropriate imprinting of certain genes is important for normal development. So that is true. Imprinting during the development process, certain cells are imprinted. So that actually fulfill the criteria of the development. But in normal adult human, there is some disease involving genomic imprinting. The name of that are Angelman syndrome or Prader-Willi syndrome. In both the cases, there is a mutation and alteration in the Angelman chromosome 15. In Angelman syndrome, maternal allele is not functioning. In Prader-Willi syndrome, the paternal allele, there is a mutation, there is an alteration in chromosome 15. Now today we will discuss this epigenetic phenomena, that means this imprinting, and how two particular genes, the name of that are IGF2 and H19, which are located 80 kb distance, and how their regulations are taking place. How their regulations are taking place. So epigenetic regulation of IGF2 and H19 gene cluster we are going to study. To start with that, you need to first know what is IGF2. You must be knowing insulin-like growth factor 2. So you know insulin-like growth factor 2, it's liver growth factor, and there are so many growth factor we have already studied. And H19 is a gene which code for an RNA, and that RNA will never translate it. So you will come across this type of RNA which are present in higher eukaryotes. Those are just present, transcription takes place. So RNA is there, but the RNA is non-functional. So H19 is such an gene who actually transcribe and will give you an RNA, but that RNA will never be translated. Now, we are going to study epigenetic regulation of IGF2 H19 gene cluster. IGF2 insulin-like growth factor and H19 genes are imprinted in mammals. This is the phenomena. They are expressed unevenly from two parental alleles. What is that? IGF2 is a growth factor expressed in most normal tissue solely from the paternal allele. So, IGF2 is functional in the paternal allele. That means in the maternal allele, IGF2 is non-functional. IGF2 protein is a growth factor particularly important during pregnancy, where it promotes both fetal and placental growth and also nutrient transfer from mother to offspring via placenta. So you can have an idea that how important it is. The parent of origin specific expression of IGF2, this IGF2 imprinting is largely controlled by allele specific DNA methylation at CTCF binding site in the imprinting control region. So as I mentioned, I will show you the IGF2 gene and H19 gene. They are 80 kb distance to each other. In between that region, one control region which is name is imprinting control region, ICR, is present immediately upstream of the H19 gene. So imagine IGF2 in the left hand side, H19 in the right hand side, center before H19, one control region is present, DNA element, that is named as imprinting control region. Some protein can come and bind. They have mentioned CTCF binding site. This is CTCF is a protein whose function is like insulator. We have just discussed in my previous lecture what is insulator. Now, epigenetic regulation of IGF2 H19 gene cluster that leads to parent-specific expression, including parental allele-specific DNA methylation and chromatin modification, DNA binding of insulator protein CTCF. Let us see what is actually the mechanism. This is a structural presentation of these two genes. Now, left hand side, as I mentioned, this is IGF2. IGF2, there are 3 promoter. We need not to know all the details. So, there are 3 promoter for IGF2. Then the IGF2 gene is there. You see 2 parallel line, that means there is a big distance between these two. Then there is an ICR sequence, which is imprinting control region. Then the H19 gene start, and downstream, very downstream, at the right hand side, there is an enhancer binding site. Okay, remember, as I mentioned, IGF2 and H19 are located approximately 80 kb apart and are expressed only from the paternal and maternal allele respectively. That means the father allele of IGF2 is working, mother one is not working. Reversely, the H19, mother's one is working, but father one is not working. Very crude way if you say. Now, little bit information. IGF2 gene driven by 3 promoters. I have shown here 2 kb apart. This side, left hand side, if you see, these are the 3 promoter are there, and then the gene are there. H19 encodes a non-coding RNA, that also I mentioned, that non-coding RNA meaning from H19 gene you will be getting an RNA, but we expect RNA to be translated to a protein. That is not happening here. IGF2 gene on the male chromosome is turned on, but the homologous gene of the female chromosome is silenced. That's what we are discussing. Conversely, H19 gene on the female chromosome is on, but homologous gene on the male chromosome is off. This is we know. Now, how to explain why it is on, why it is off? Come to the next slide. We'll try to explain. Very simplified model of IGF2 and H19. This is maternal allele, which is of white color, a cream color. The green color is the paternal allele. So we are seeing 2 chromosomes here. Okay. So one is from mother, one from father. So in the mother, what is the structure? You just see they are 80 kb apart. That's what they are showing. IGF2, H19 is orange color one. In between, one ICR imprinting control region is there. That is in the gray color. You can see, and I told you far down there's an enhancer. Now in the maternal allele, what is happening? Look at very carefully. This imprinting control region, there one protein, this CTCF protein, is a known mammalian or eukaryotic mammalian protein which can act as an insulator. So this can come and bind here and protect the enhancer who is far downstream this side to exert its effect to IGF2. So that's the reason the maternal allele of IGF2 is not functional. At the same time, H19 from the maternal, from the mother allele is working because it is, the insulator is before that, so it is not inhibiting its effect, but the insulator is inhibiting IGF2 gene expression. Come to the paternal allele, the same thing is happening. Now in the paternal allele, some other modification takes place. What happened? The paternal allele is methylated at imprinting methylated ICR. That means this methylation silence the gene. Methylation will takes place and it will form an heterochromatin formation. Which region is been methylated? Just before the H19. Now H19 is non-functional because that imprinting control region is now methylated. Remember, this is an imprinting site. This is the locus where methylation takes place, and if the methylation takes place, which is very much nearby, this is very much nearby H19. H19 is silenced because of this this methylation, and because of the methylation takes place, so CTCF protein cannot come and bind at present. So enhancer, even though it is present nearby of H19, it cannot exert its effect because it is silenced already, but it can exert its effect to the far distance IGF2. So IGF2 is working. So this is the explanation behind why the paternal allele of IGF2 is functional and why the maternal allele of H19 is functional. Read carefully, try to understand the mechanism behind here. The last slide is actually explaining everything. Hope you will understand. In case any difficulty, call me, I will explain one more time. Thank you very much.